We'll be moving on to our next session on spatial transcriptomics, and our first speaker is Chen from MD Anderson Cancer Center, and he'll be telling us about spatial gradient analysis for spatial transcriptomic data.
Okay. Thank you. Can you hear me? Okay, I'm here to share one of our recent work is to detect spatial gradients from the transcriptomic data, spatial transcriptomic data. And this is my postdoc work, one of my postdocs work. I guess we're pretty much motivated by the biology. And as you know, the kind of spatial gradients, including the morphogene, they kind of direct the patterning of the cell distributions from embryogenesis and organogenesis. And in cancer, it's kind of a little more complicated, but I guess we have been inspired by the earlier talk, like especially the talk in granuloma. Quite a few work has referred to that. And there's a lot of metabolic and also cytokine gradients that is actually existing in tumor tissue, and they have uneven distribution at different location of the tumor. Therefore, I guess they are kind of they basically create a lot of different stimuli to the cancer cells, and also immune cell could infiltrate into different parts of the tumor. So those factors actually driven many different type of potential gradients that exist in the cancer tissue. So our goal is more kind of a discovery of those potential gradients from spatial transcriptome. So, and I think this has been covered quite a bit, so I'm not going to into the details. In terms of analytic approach for spatial transcriptomics, there has been a lot of being developed, but we think, rather than the segmentation and also annotation, I think there's a relatively less, addressed is about this type of organization, high order organization that is driven this type of uneven distribution of different programs. The earlier work on spatial variable gene statistical tests, those were focused on individual genes which were expressed according to some spatial coordinates. We think it's probably less effective in the spatial transcriptomic data right now because the data is very sparse. And oftentimes when you have kind of a spatial gradient, they are not affecting just a single gene. It's potentially affecting an entire pathway or combination of pathways. Therefore, we think it's more effective to actually try to detect those pathways as a gradient. So, of course, there has been a lot of work in terms of identify pathway and gene sets in that space. Particularly, people has been using non-negative matrix factorization on the transcriptomic data to identify the so-called gene programs, which are, basically partitioning molecular data into different kind of a component or factors. So we are, our basically our thinking is we can apply similar negative matrix factorization techniques on the transcriptomic data, and then we try to project those components. We try to do a simple linear regression of trying to predict the distribution of those components from the spatial coordinates, through this linear regression, as you can see here. And then we identify components which actually have pretty much well predicted by those by the coordinates. And through this, we're able to actually identify, kind of where the particular, there's a particular activity of a component and in what direction this component is actually projecting to. So that way we actually identify both. We kind of create this spatial tomography, whatever has been used, but we're kind of focused on where the slope is very deep, and we're able to actually identify the kind of a boundary of those, in this manner. And we started, particularly interested and actually followed the earlier work by GBM. Dr. Tirashi and also Suva has been looking at the GBM tumor for quite a few years. And they are able to actually from transcriptome to deconvolute the programs into those four major, kind of a cell states. And also they recently published and they found the hypoxia or oxygen level is actually a very much driven factor of spatial organization. When there's less of oxygen, like hypoxia abundant tissue, you saw a lot of organized, layered, configuration of the tumor. But in places where there's lots of oxygen, you actually saw those cells actually is kind of disorganized. You can't actually saw much of a gradient in those places. So we want to use this as a kind of our kind of a proof of a concept, positive control to look at the spatial gradients detection. So this is one example we were able to generate at the earlier phase. This is one of the GBM sample. We were able to project those NMF components into the space, and the arrow actually pointing to where the activity of this program is relatively high. And as you can see, if you run this program LSGI, you will get a lot of arrows on those. This is 10x wisdom data. This is actually kind of a reminiscent to the RNA velocity, although now we're kind of looking at into the space. And if you do some spatial proximity analysis, you can see some of the programs actually located pretty close to one another. For example, in this example, you see program 2 and 4 actually very much coincide, localized to those locations. And just to extract those two components here as red and kind of greenish, you can say, interestingly, they were actually, kind of opposite pointed, they're kind of back to back pointing to two different directions. One of the program, actually, if you do the annotation, you find out they're actually the assay-like states. And the other program is actually the hypoxia, states. Basically, this is the mass-like states. So, as you can see here, we already find out there's a kind of a sharp boundary between those two programs, right at the end of those, kind of intersections. So this is kind of reminiscent of what granuloma talk, was given yesterday, even though this is cancer tissue. You can see there's a kind of a sharp change from a switch from program to other programs. And our program is able to kind of give you some indication of where they are and how they distributed in the tumor tissue. Okay, so we then looked at the tons of 25 actually GBM samples and ordered them by the hypoxia abundance, which can be detected based on the mass-like states of those programs. And then we actually calculated how many gradients component, NMF component we can detect. And we kind of correlated the amount of NMF spatial gradient versus the hypoxia level. As you can see, there's a nice linear correlation. So this is a kind of validation of we are actually detecting the known hypoxia-driven factors in the organization of GBM tissue. And then we kind of scale up this into a pan-cancer cohort where we look at 87 tumor, including 14 different tumor types. We try to analyze each sample individually, and then we kind of try to cluster them together to derive so-called meta programs, similar to their early work. And based on those meta programs, as you can see, we kind of identified 20 different meta programs, and a bunch of them actually appear to be pan-cancer programs, and some programs are more cancer specific. And if we annotate those, we can see the PanCamp programs is actually well-known, EMT, OX4, and also some immune reaction programs are involved. But programs like hypoxia, they actually were specific to certain cancer types like GBM. They are not actually universal programs. So this, makes you wonder, what are the specific programs in each, those cancer types can be identified. So one of those we found is actually the TNF-alpha and NF-kappa B program. Seems to further delineate the GBM into, different, kind of a spatial organization pattern. For example, as you can see here, this program is helping us highlighting different pockets of tumor cells. They were actually having high TNF-alpha and NF-kappa B activity, potentially has some immune infiltration there into those pockets, but it's difficult to see this from the wisdom data. So then we further actually apply this technology into our own data. This is a mesothelioma tissue sample. Mesothelioma, as some of you know, is a type of tumor for individuals unfortunately exposed to asbestos. And this is basically tissue from lung, which is a biphasic type of mesothelioma where you have different histology in the same slides. And our pathologists were able to actually perform very deep annotation of different histology subtype in those tissue. And we're trying to see how well we can look at those from the spatial transcriptomics. And, if you perform spatial domain analysis, pretty much you can isolate or you can segment the major, regions of those slides. And if we perform our LSGI analysis, now in addition to those domains, we actually have lots of arrows which help us not only further indicating where the boundaries are, but also help us understanding what are the programs is driven those difference in the histology. So if you look at the particular programs, we have be able to isolate, and as you can see, very interesting. For example, EMT program is actually driven a lot of spatial heterogeneity actually across disjoint area on the slides. You wouldn't know they are actually driven similarly by EMT without actually perform this type of kind of projective analysis. So then we feel like the resolution of VISM is actually kind of limited. So we try to apply this to Xenium data, which is not whole transcriptome, but you still got a lot of information to perform this kind of a spatial transcriptome gradient analysis, although it's more kind of a gene-based instead of a pathway-based analysis. So on this same mesothelioma slides, we identify one of the FOVs, and we're able to create cell segmentations, and we are able to project the kind of a spatial gradient at single cell resolution to those FOVs. And as you can tell here, actually, we're still kind of look at those as we're speaking. But interestingly, the major programs identified, for example, this y-axis blue program, the top gene is actually IDO1. I just actually found this very interesting because that's also what Aaron talked about yesterday on granuloma. IDO1 gradients is actually driven a lot of spatial organization of those. Maybe it's shared between cancer and granuloma. Who knows? Other program is actually more of a tumor-specific, program with expression of gene like mescaline genes here. So we're to some degree already be able to kind of identify the gradients at a kind of a single cell level to help us even further decompose the tumor tissue into different pockets of activity, which help us understanding the particular resistance of the treatment, or maybe there are therapeutic opportunity involved in looking at those data. So okay, with that, I just like to quickly acknowledge people who has done the work. First, my postdoc Chen Liang, who's the one who develops the program, and he actually is on faculty job market now. If you're interested in talk to him. And Najla, who's the one who has been driven a lot of analysis of our, facilitate our collaboration with our translational molecular pathology program, led by Kara and Luisa here. They are both, all of them are here. Should talk to them. And Najla has a poster this afternoon describe we are exercise or compare different spatial platforms, transcriptome platforms. And if you want to know the technology, it's very good to talk to Najla. And then this is a large collaboration between the bioinformatics and also the pathologist team at our institution. And we are trying to develop the capability to analyze the data. We're also kind of learn a lot from the conference, and very much thanks the organizer to give us opportunity to share our work. And I feel very much connected to you guys scientifically. Like I talk about granuloma, but I feel a little intimidated by large language models. So therefore, I presented something relatively simpler. Hopefully, it's easier to understand. Okay, with that, I'll stop here and welcome any question.
We have time for one quick question. Anything on Zoom?
Hi, that was a really nice talk. So I had a question about the spatial gradients and what kind of, I mean, maybe more of on a broad level, what kind of information can we kind of glean from that? So this is a pathway analysis. You have some directionality that's going on. Should we make some conclusions about cellular migration, or are we making some conclusion about upregulated pathways that are going on? And I'm just trying to better understand like what some of the flow actually is referring to from a biological context. Thanks.
Yeah, I think migration is potentially a little difficult because that requires temporal kinetics. Right now, we're just looking at static image, this program. So I think it's more of a segmentation tool at the pathway level to some degree to tell you where the activity is high or low and where the boundaries are. And one of the analysis besides NMF, you could also use your hypothesis. For example, you're interested in particular metabolites or pathway. You could basically project that type of gene sets on the spatial map to say where they are. I think good news, I think spatial transcriptomics, even though it's low coverage, low resolution right now, it already can help you kind of do those hypothesis generation on the data. So that's where we're kind of coming from.
Okay, thank you so much. Please join me in thanking again Ken for his talk. Up next, we'll have Izabella Zamora from the Broad Institute speaking about PlastiNet.
Let's see. Okay. Oh, that clicks. Cool. Okay. Hi, my name is Izabella Zamora. I'm currently a computational associate in the Hacohen lab at the Broad Institute. And today I'm excited to share PlastiNet analysis framework we've been working to develop to better understand cancer cell plasticity. Specifically, our goal is to explore the epithelial to mesenchymal transition by applying graphical attention networks to spatial transcriptomics data. To give some context, EMT is a critical form of cancer cell plasticity, which we define as the ability of cells to transition between different states. EMT is central to tumor progression, metastasis, and therapy resistance, making it an important target for therapeutic interventions. In this presentation, I'll focus on how extrinsic factors within the tumor microenvironment drive cancer cell plasticity, particularly in PDAC. To investigate these extrinsic factors, we leverage spatial sequencing data, specifically CosMx, which provides single cell resolution. However, it has a limited gene panel with only 960 genes. It can be challenging to finally resolve tumor substates. And to address this, we propose that the spatial context of a cell can fill in the gaps, allowing for better identification of cell states. And by analyzing the spatial data, we can also understand how the cell's environmental influences change these states. Ultimately, our goal is to identify potential targets for immunotherapies and combination treatments. The framework I'm presenting today is based on 3 key assumptions. 1, that cells physically close to each other are more likely to be clonally related than cells that are super far apart. 2, the direct neighbors of a cell provides valuable information about the cell's state. And 3, EMT and PDAC follows a linear trajectory from epithelial to mesenchymal states. The first step in this process is to construct a graph from the spatial data. We start with lots of cells and their XY coordinates. We then define a radius that captures direct neighbors of a cell, where any cell within that radius are connected via an edge. In this graph, each cell becomes a node with its gene expression as its feature space. And for each edge, we calculate the distance between neighboring cells. So in summary, cells are nodes, their feature vectors are the gene expression, edges connect neighboring cells, and the distance values are recorded between these cells. This distance will later be used to weight how strongly neighboring cells influence each other, with the influence decreasing as distance increases. Now that we have a graph representation of our tissue, we feed it into PlastiNet, a deep learning framework built in the form of a graphical attention network. In this simplified example, we have 3 cells, the target cell highlighted in blue and 2 neighbor cells in gray. And for each target cell, the model looks at both the target's own gene expression and the gene expression of its neighbors. The goal is to compute a weighted sum of these influences, which is used to create an embedding that captures more than just its own gene expression. And these weights are denoted as A and tell the model how important each feature is. Initially, all of these weights are uniform, meaning all genes are considered equally important from neighbors and self. The model is trained over many cycles using a loss function to adjust the attention weights. This loss is a sum of 3 different components. The first one is Deep Graph Infomax, DGI, which helps the model learn useful representations of cells by encouraging it to capture patterns in the data over randomness. The second one is a spatial loss term, which ensures that cells that are physically closer to each other have more similar embeddings than cells that are super, super far apart. And an L1 loss term, which encourages sparsity in the attention weight, so the model focuses on the most important genes. Once a loss has converged, the attention weights are considered optimal, and the model combines the gene expression data from each cell with the attention weights, creating a final embedding space denoted as Z. The embedding space represents each cell in a way that captures its gene expression, its relationships to neighbors, and relationship to the graph as a whole. This learned embedding can then be clustered, where each cluster represents a repeating spatial pattern in the tissue, also thought of as neighborhoods. And we can also order cells of interest along a continuous trajectory using a walk-based pseudotime approach. In this case, we're looking at epithelial cell differentiation along the EMT spectrum. And finally, the attention weights learned by the model can provide insights into which genes or features are most important for defining relationships between cells. And these weights can be analyzed to understand the key drivers and learned tissue structure. So to summarize, PlastiNet creates a spatially aware embedding using an attention mechanism on a graph, which can then be used for downstream analysis, in our case, to better understand extrinsic factors of EMT. To validate our approach, we applied PlastiNet to a well-studied system, the colon, which has well-defined spatial and transcriptional differentiation paths. Cells move from the crypt, where the stem cells reside, up to the villus, where fully differentiated cells are shed. When clustering the spatially aware embedding into neighborhoods, we observed distinct layers across the tissue, the bottom layer representing the submucosal, followed by the stem cell crypt, and so on. Each layer is composed of different cell types, giving each neighborhood a unique composition. The clustering resolution can be adjusted to capture broader or more refined neighborhoods, depending on the analysis needed. Next, we estimate the differentiation path of the epithelial cells, applying pseudotime to this embedding, and find that it accurately reflects the expected stem to differentiated cell trajectory spatially. And to validate this transcriptionally, we checked key marker genes and found that stem markers were highly expressed at early pseudotime points. Transitionary marker CD24 appeared in the middle, and differentiation markers CDH1 and KRT20 were expressed at later time points. This helped us tune our model approach and confirm that we can capture biological neighborhoods and differentiation paths spatially and transcriptionally. We next applied this to a more complex spatial graph, PDAC, where our differentiation path of interest is EMT. We use CosMx data from primary untreated PDAC samples, courtesy of the Ting lab. The clustered results from the embedding correspond to these distinct tissue structures, which were labeled based on cellular composition. To focus on a couple key neighborhoods, these are our immune-heavy neighborhoods. There was a global immune region composed of a mix of different immune subtypes, a cancer-associated fibroblast-enriched neighborhood highlighted in orange, and a potential immunity hub in dark gray, which was macrophage and T-cell rich. We also identified 2 distinct tumor neighborhoods. One was predominantly composed of tumor cells, and another was mixed with immunosuppressive cell types, such as CAFs and SPP1 positive macrophages. The immunosuppressive neighborhood spatially co-localized with a CAF-enriched immune cluster, reinforcing the hypothesis that tumor cells in this neighborhood exhibit potentially more mesenchymal-like local environment, while the pure tumor neighborhood, which formed more ductal structures, appeared to be more epithelial-like. And this suggests a potential separation of cell states at the neighborhood level. We then constructed a differentiation path using classical and basal signatures from literature. The continuous cell ordering reflected the expected expression patterns. Epithelial genes were highly expressed at early pseudotypes and decreased over time, while mesenchymal genes increased later. This was also morphologically consistent, as low pseudotime cells, the more epithelial-like, form structured ducts, and the high pseudotime, the more mesenchymal-like, were more dispersed throughout the tissue. And we validated this with spatial autocorrelation as well, which I'm not showing. So in the interest of time, I will skip to our more interesting conclusion. And that from our ligand receptor results, we found that CD44 had increasing expression along the EMT trajectory, and it had different binding partners across the EMT spectrum. And upon reviewing the literature, we discovered that CD44 undergoes a state-specific isoform switch, adding extracellular exons in its variable form. To investigate this further, we used RNA-seq and mass iso-seq data from patient-derived PDAC cell lines. This data was generated by others at the Broad and allowed us to explore the CD44 isoform switch in more detail. Labeling cells based on their RNA-seq expression revealed a clear separation between the epithelial and mesenchymal states. Our analysis identified that the standard CD44S and a variant with additional exons 8 through 10 were present in our data, where CD44S was the most common form, but the variant was mostly in the epithelial states. CD44 expression was also overall higher in the mesenchymal compartment, and we saw ESRP1, which is a known regulator of this isoform switch, expressed in the epithelial population. So we show that CD44V isoform is primarily present in the epithelial states, and in this specific cell line, it included the exons 8 through 10. One of the CD44 ligands of particular interest was SPP1, which is known to have negative effects on survival and overall tumor progression. We wanted to explore the different CD44s exhibit any preferential binding to SPP1. So we simulated the binding complex using AlphaFold, and we found that the variant isoform predicted to have stronger binding with SPP1 compared to the standard form. And looking at TCGA data revealed that higher expression of this isoform exons correlated with lower survival compared to lower expression of these variant exons. And while these results are very preliminary and require further validation, they offer motivation for a deeper investigation into the role of CD44 isoforms in tumor progression and survival. In conclusion, I hope I've convinced you that this framework can be used to better understand spatial sequencing data. And although much work is left to be done, we have been able to reconstruct cellular differentiation paths in normal colon tissue, as well as apply this to tumor tissue to identify neighborhoods, as well as resolve a spectrum of plasticity states. Through this analysis, we identified CD44 SPP1 axis as a potential important mediator of tumor cell plasticity, specifically in the isoforms, and with preliminary evidence suggesting preferential binding and impacts on survival. We're still working to better interpret the attention weights to gain insight into what defines these patterns that the model has identified, additionally validating the spatially or isoform ligand receptor relationships, applying long-read sequencing to Visium data, and explore other interactions and niche factors along this EMT spectrum. So with that, I would like to say thank you to everyone that made this possible, members of the Hacohen and Lander Lab, my mentor Arnav, as well as all the wonderful people across the Broad, funding sources, all the patients and their family, and thank you for your attention and the opportunity to present today. Hopefully we can get to lunch. There's not, sorry for cutting in.
Okay, we can take a few questions.
Okay, great talk. 2 questions from Zoom. The first one, I think you mentioned in like the last bit when you were talking about future directions, but the question is, can this model be applied to Visium spatial transcriptomics data, especially the more recent Visium HD data?
We have not tried to. I think with the number of genes in Visium, the runtime would not necessarily be conducive, but it is definitely something that we can look into adapting for.
Awesome. And then the second question is PlastiNet able to integrate multiple single cell spatial transcriptomic samples for identifying niches, and is it released?
It is not released. We're working on putting the code together in the next couple of months. As far as like multimodal integration, we do expect that the feature space is pretty flexible. It's just a vector and we can append onto it, but we have not necessarily tested it.
Okay, thank you. A great talk. I'm interested in the way you build the graphs for the post analysis. And I think the first step is pre-process and do the segmentation of the cell and then build a graph based on the segmentation result. Is that correct?
Yeah, so we use the segmented data to construct the graph.
Okay, so after you train the model, I think you can visualize the graph attention weight to review further interpretation, right?
Yeah, we are working on that actively right now.
Okay, sure. Thank you.
Okay, thank you. Okay, to start us back on the spatial transcriptomic session, we have Roy Maimon from Don Cleveland's lab at UCSD, and he'll be talking about generating new neurons in the aging brain.
All right, can you hear me okay? Awesome. So yeah, thank you so much for having me. It's a great opportunity. My name is Roy Maimon. I'm a postdoc with Don Cleveland at UCSD, and today I'm going to share with you our ongoing project on how to generate new neurons in the aging brain. And I would really like to start with this fascinating animal, the axolotl. I hope you know it. For me, it's very inspiring because you could really take half of its brain out and it will fully regrow. Unfortunately, it's not the case in mammals and in human. And as Ramon Cajal, the father of neuroscience, stated in 1913, the neuronal paths are something fixed and immutable. Everything may die, nothing may be regenerated. And that's why neurodegeneration is known to be irreversible. However, Cajal also stated that this is the duty of next generation to find a way to overcome the intrinsic failure of the brain to regenerate. And here we are today, not quite next generation, more like 4 or 5, I would say, but we are trying to tackle this down. So let's say we want to tackle Cajal's challenge, generate new neurons in the brain and replace dead neurons. How are we going to achieve it? So there are 2 ways to go. You could either inject iPSCs or neurons in the brain to reinnervate the brain region, or convert glia in vivo into neurons to achieve this replacement of dead neurons. We're trying to do both in our lab, in the Cleveland's lab, but today I'm going to share with you how we're trying to convert glia into neuron in vivo. So let's say we want to do it. How we're going to do it? So I brought you just a very simplistic view of development. So there are radial glia cells that converting into neurons in the developing brain. And if you look, again, this is very simplistic view. There are 2 loops. So there are silent transcription factors, neuronal transcription factors here in red in this glia. And there are some other genes that further suppress it down, such as PTBP1 and REST. And we will show you them before. And then upon conversion, so these switches are turning up and down, so switches going on and off. And so you start seeing expression of neuronal transcription factors here in green, and the barriers, these gatekeepers, are gone. And this is basically what we are trying to mimic in the lab. So we are trying basically to mimic development. Now, how are you going to do it? So there are many ways to go. So most of the groups are trying to do it with lentiviruses approaches, either overexpress of some genes or remove the barriers with short hairpin. But we are trying to do it in more therapeutic viable approach, the antisense oligos. So these are basically small DNA drugs that you could design. There are 20mers to target any gene that you want to knock down. And it's been shown before to be it's FDA approved drug, so it's safe. And if you want to generate new neurons in a therapeutic viable approach, we think that this is probably the way to go. So we use these ASOs to target one of the genes that I showed you before, PTBP1. So this is an RNA binding protein, best to know working with the spliceosome. And it's been claimed before to be part of this glia into neuron conversion, at least astrocyte into neuron conversion. So we want to understand whether we could design ASOs to target PTBP1 and generate new neurons in the aging brain. This was the idea. So we designed these ASOs with Ionis Pharmaceutical. This is their bread and butter. And we screen these ASOs and we deliver them all throughout the CNS or the CSF. Wait for 2 months and look what is happening. So at first, in 2010-21, we used genetic approach to really label permanently glia cells in red, with the idea that if they're convert now, so we'll start seeing neurons or neuronal morphology that are red and colocalized with neuronal markers, as you could see here. The only brain region at that time that we saw generation of new neurons was the hippocampus. So this is the hippocampus is the brain. I'm sorry, I'm talking about the brain. No one here is brain, but let me know if you need. But this is the dentate gyrus, and you could see all these yellow new neurons. These are yellow new neurons that we generated in this brain region, and we think we could generate 4% new neurons at least at this brain region at that time, and it's already published and it's characterized. I think it's really interesting, but one of the highlights is that we think that these 4% of neurons could really modify mouse behavior. So when we test the memory of these mice, we see that they are behaving better in Barnes maze, so they find a way out faster than the control group. So this was interesting, and we concluded at that time that ASO-mediated transient suppression of PTB lead to the appearance of new neurons in the dentate gyrus, and here you could see one example, control versus the ASO injected at least 2 months after, but there are many open questions still. We don't know what are the cell of origin. We don't know what are the intermediate state and what neurons they're becoming, and these are only some of the questions. And also it's very exciting times because there is an active debate in the field. So other groups try to convert neurons using this approach, but couldn't. So we really want to shed more light on these questions and this controversy. And we thought the best way to go, for us at least, was utilizing single-cell spatial transcriptomics. And this is what I want to share with you today. Specifically, we are using Murphy's approach. So this is this approach invented in 2015 in Shawi Zhang's lab at Harvard, 2015. And the idea is quite simple. It's more complex. We could discuss it later. We all know what it is probably, but it's instead of you doing single molecule fish and image 4 genes at a time, we could do up to thousands of genes. And this is due to combinatorial labeling, sequential imaging, and error-bus barcoding. And this is what we're doing. And luckily at that time, Professor Bogdan Bintu have joined UCSD, so he's 2 stairs behind us, and he helped us to build this custom-made Murphy scope that is very similar to VisGen that you probably all know, but is custom-made. It's not as pretty as VisGen, I would say, but we think that it is doing better job. So when we calculate the transcript per cell and compare our custom approach to all other commercial approaches, including Xenium, Cosmix, and Merscope, we see 10 times better transcript resolution. There is also other advantages that we could discuss later, and I'll just we could basically discuss later, but we really think that this is really highly stable approach because here you see one example that we image 2 genes, NR and GN in red. So each dot here is 1 mRNA molecule in a different cell, and GAD1 is in green. And we perform 74 hybridization round, and we perform the same gene imaging at that time, and we see that there is 99%. So it's indistinguishable. So the approach is highly stable. And we think that it's because we improve it with a special protocol that, again, it's going to be published, and we could discuss it later. And we name it Murphy's Plus. And stability is only one advantage, but we also think that we have higher throughput. So instead of imaging 3 brains at a time using VisGen, for example, we could do 3 times higher. And we called it Murphy's Plus, and again, everyone will be able to use it. And we use this approach to generate what you're all familiar with, the 2D UMAP space, and we project it back to the brain to really identify all the cells and the clusters that composing the tissue. So again, this is coronal section of the mouse brain, 8-week-old, and we image here 60K using Murphy's Plus, and we imaged 2,500 genes at that time. Now, all this information is going to be uploaded online, so everyone in the community could use it. We name it Fisheye. So this is online visualizer that KJ, really great undergrad, have built, that everyone could use it. So all the data is going to be there. You could look for your favorite cluster, your favorite gene, and really look in early development and even in aging mouse brain, what is happening in these 2,500 genes. This was the idea. Now, using this online visualizer first, because we want to knock down PTBP1, we want to know where PTBP1 is actually expressed. It wasn't known. So here you see one sagittal section of the brain. It's P5. It's very early in development. And in red, you see cells that expressing more than 35 mRNAs of PTBP1, and blue is 0, and this is the scales here. And you could see that is ubiquitously expressed, at least at this age. However, when we look, to our surprise, in 1-year-old, we see that it's way more confined. So this is the same scale, but we start seeing that the PTB is actually confined to the subventricular zone. So this is the ventricle. It's another neurogenic niche. And you see that PTB is highly expressed in the choroid plexus, which are these cells here in the middle, and ependymal cells, lateral ependymal, and importantly, also in progenitor cells here in yellow. And we think it's important because when we deliver the ASOs, wait for 2 weeks, we really saw reduction in all the ventricle, but specifically we saw reduction in PTB and its target only in progenitor cells. So it narrowed down our investigation to these progenitor cells. Now, is this reduction in PTB meaningful? We really think so. So here you could see again our merfish attempts in 8-week-old. So we capture again the subventricular zone. We see in green here ependymal cells lining the ventricles. We see the progenitor cells in yellow, and we see new neurons, new immature neurons. And this is active neurogenic niche, at least in young animal, it's still active. However, when we did the same experiment in a 1-year-old where there wasn't supposed to be any immature neurons, we really don't see much. But we still see these dormant progenitor cells. Now, 2 weeks post-injection of the ASOs, we perform the same experiment, and we start seeing the reappearance of these immature stages of neurons in both neurogenic niches. And when we compare the transcriptional space of these progenitor cells, the activated one, and the dormant, we see that they are different. So these are activated progenitor cells starting express markers of immature neurons, so DCX, OX11, etc. And to our surprise, also markers of proliferation. So you see that we generated new neurons, but the pool of the yellow cells, we didn't replenish them. We replenished them. We replenished them. So we took advantage of this feature of proliferation and decided to deliver EDU, which is a marker for proliferation. So it's basically analog for thymidine that we could then look and see which cells are now proliferating. And the idea was to do it in all our conditions and couple it with merfish back again to identify which are the cells that were proliferating and what they're becoming. This was the idea. And here we could see one example of a sagittal section. This is a very young animal, 4-week-old. And each dot here represents one cell that undergone division. And you see that the EDU is incorporated all throughout the nuclei. So it's really consistent with proliferation rather than DNA damage. And we confirm the appearance of these cells in the dentate gyrus, one neurogenic niche, in the subventricular zone, the second one, and in the olfactory bulb, which is really what others have already established. But we did it in the 1-year-old control and in the PTBSO and coupled it with merfish. And in the 1-year-old, we don't see much. So there isn't a lot of proliferation and division in the 1-year-old, just naive conditions. However, when we deliver the ASOs and we look 1 month after, we start seeing all these progenitor cells that were dividing probably, and then the immature neurons, some glia, but importantly also new neurons that were generated and penetrated into the striatum. So this is one brain region called striatum. And you see all these yellow dots are new neurons that were divided upon PTB suppression, penetrate there and become neurons in this brain region. Now we also did sagittal sectioning and cross-sectioning to validate it in many animals. And we counted around 1% of new neurons in this brain region, which we really think is important and could, again, modify mice behavior. Lastly, what we did is perform this experiment in a human context, in a human brain. So here you see merfish experiment in the human developing subventricular zone. Again, this is the same brain region we were focusing on. And we performed 1,200 genes merfish. It looked pretty good. The RNA is really high quality. And we performed this, again, the same pipeline, 3D UMAPs, and then brain projections. And we identify all these progenitor cells that are active and generating immature neurons, stage 1, immature stage 2. So all the stages of immature neurons. And we have a pretty good understanding of this process now because of this approach. And we know where they're going and what genes they are expressing. And again, this is going to be available to the community. To our surprise, also when we did the same experiment in the aging subventricular zone, so this is 70 years old human tissue, we see that the progenitors are still there. So these are the progenitor that we can convert, but we don't see any new immature neurons. So this niche is, again, is quiescent. However, when we look on PTBP1 expression in these tissues, we see that the cells that highly express in PTB are progenitor cells. So to me, it's like these cells are just waiting for us to be converted. So hopefully in the future, we will have something in this front also. So altogether, I just will summarize and say that using spatial transcriptomics, we try to understand this glia interneuron conversion or generation of new neurons, and we identify an active neurogenic niche in a 4-week-old mouse brain that is quiescent in the aging brain. And upon PTBPASO, single injection of this molecule into the CSF, these cells are now dividing, and we could reactivate this neurogenic niche and generate new neurons. And it seems that these cells that are converting are also in the human brain. And I will also say my vision a little bit. So hopefully in my own lab in the future, in the near future, what I really want to do is to use this approach, glia to neuron conversion, and apply it in neurodegeneration. So first, I want to start with Huntington disease, which Sonia Vasquez-Sanchez, she is also here, and she'll give a talk. You'll hear more about it, and see if we could generate new neurons in neurodegeneration. I also want to study it in a human context. So I want to generate a model that we could then study conversion in human context. And I want to identify novel reprogramming factors using approaches that we heard here before, Pertapsic and other, and use Murfish as an output. And that's it. With this, I want to thank my beautiful lab, Don Cleveland and Bogdan Bintu. All this work is shared with Carlos Tion Marinas, Sonia, that she is also here, and all the other team. And yeah, thank you, and I'll take any question. Thanks so much.
Very cool. So on Murfish 2 plus
or jellyfish.
Plus.
We're still, we don't know. But
I like jellyfish. Yeah. So I guess the question I had was whether you can elaborate on like where the enhanced sensitivity is coming from, and how the higher performance, to the extent it is affected, does it impact? Because I know with a lot of these assays, especially the commercial ones, right, at least talking to some of the scientists there seems to be this Goldilocks zone they're trying to keep on a per transcript level so that they don't get spot coalescence to confound the IDs.
No, definitely.
Right. And so how does,
right,
I guess one is like, where's the sensitivity, where do you feel like the sensitivity is coming from? And then two, with that extra sensitivity, how are you able to maintain the same dynamic range or avoid those problems?
Yeah, I wish I had a slide. I might have, but never mind. But the idea, it's quite simple. I think others also using Lonkai. I think also using a gel, casting gel. I'm not sure, but what we are doing is designing our probes. So each probe of us, it targets the mRNA, and on the N terminus of the probes, we have acridite modification. So it's acridite, and then upon hybridization, everything is there. We're casting acrylamide gel. So basically, the probes are just sitting in a gel. And even with this harsh hybridization round, the RNA is probably degraded, but the probes are in set. And we don't care about the RNA anymore. So then it gave us really, we went up to 200 hybridization round, and Sonia also developed protein stainings and DNA staining, and it's working pretty well. So this is where it came from. We also improved the cell segmentation and Merlin, so it's a bit Merlin. So when I say we, it's mostly Bogdan Bintu. So I'm just a little pawn, but yeah. So this is what we think is giving us the more sensitivity and stability.
Do I have time for one more? I'm looking at Anna because I'm not sure. I know Kelly's in charge, but okay. So more about the biology, so the cool phenomena. So obviously, like, there seems to be, even in aged humans, progenitor looking cells still there in the brain. This PTB seems to be an important regulator that's holding these progenitor cells into like some sort of quiescence, wait homeostasis mode before kicking into regeneration. So obviously you've synthetically been able to perturb that and activate them. What do you think more broadly is like the biological evolved mechanism that this is normally in place for? Like, so what is this there? Is this part of a sensing mechanism? Is this like why are these cells still there? Why is this pathway in place?
Yeah, this is, I love this because, so it's definitely because of something due to development, right? So we see that this process is active. So PTB is up in these progenitor cells during development, and then you see its reduction, and then these neurons becoming neurons naturally. And then with aging, it seems that we're losing this ability for some reason, unless I think also if you, we see also if you perform TBI, for example, or other injuries, so these progenitor cells maybe then will reactivate it. But other than that, there aren't. Now, this is also interesting because, for example, the axolotl that I showed at the beginning, so there it's active. So this same process is active. Why did we lose it, humans? And there are many interesting theories. So some would say because humans need to run from the predator and the blood will go. So there is a lot of theories, but it's unknown. I think the main theory is, yeah, humans had to run from predators while trees and axolotls not running. So, and if you run, the blood will just spill. Or so this is why the regeneration doesn't happen, and you have you're forming a blood cup instead of regeneration. So this is one theory, but it's unknown basically. But we lost this ability for some reason. The cells are there, and unless activation, they won't go. Yeah.
Okay, join me in thanking Roy. Next, we have Emily Thomas speaking from Oxford on spatial mapping of human bone marrow.
Well, thank you so much for inviting me to give a talk today. It's really great to be here. I'm Emily, like was just mentioned. I'm in the Royston and Rittscher lab at the University of Oxford, and I'm going to talk you through today about our bone marrow spatial transcriptomic dataset and how we've incorporated our previous work integrating H&E-based ML algorithms to better explore and augment our dataset and show you a couple of bits of preliminary analysis. So our lab works on a type of blood cancer called myeloproliferative neoplasms, which are characterized by overproliferation of a type of blood lineage. So patients present with one of four subtypes, so ET and PV, which you can see on the left here, which can potentially progress via pre-PMF to myelofibrosis, which is characterized by scarring of the bone marrow tissue. So to diagnose and classify these MPNs, pathologists will use a bone marrow biopsy stained with H&E, like we've seen many, many times, and then they'll produce a qualitative report. But the trouble with this is that it's inherently subjective. So a major unmet need in MPNs is to be able to have a quantitative way to classify the different subtypes, know which patients are going to go on to clinically progress, and assess the impact of new therapeutics. So given this need, our lab has already started to try and quantify the bone marrow environment using routinely available sections. So we focused on two key features here, so fibrosis grading and then megakaryocyte detection, segmentation, and phenotyping. So megakaryocytes are these big, well, potentially big platelet producing cells in the bone marrow, and they're a commonly used marker when we're trying to classify MPN subtypes, and they're also implicated in fibrosis. So from this work, we've managed to phenotype them into 9 distinct phenotypes, and I'll talk a bit more about them later. But that's just 2 views of the bone marrow. So the obvious then next thing to do was to extend this to spatial transcriptomics, and we've chosen the Xenium platform to do this because it gives us a ultra-fine view. We've got the cellular resolution. We are only using the 377 pan-cancer gene panel, but importantly for us, we can then have the same slide which has the H&E, so we can transfer our ML algorithms from there. So this leaves us with this nice, beautiful data set of 31 bone marrow samples and spanning the whole range of the MPN subtypes, and we've got 5 normal samples as well. So the next thing to do was to develop a pipeline to be able to make the most of all of this analysis. So I'm going to talk you through each steps of these, and these are some of the packages that I've found most helpful so far. So the first thing to do was, given their importance in clinical assessment, was to be able to make sure that the megakaryocytes are accurately captured. So the in-house Xenium package does a really good job at segmenting the, wherever there's the DAPI stain, segmenting it. And then doing a Voronoi partition of the space. But the trouble with this is that megakaryocytes are often polylobated, and given we're really interested in their clustering, we want to be able to capture them properly. So running our MEG pipeline on the accompanying H&E, we can just import them into our segmentation, and then we can also have their phenotypes, which we can correlate with gene expression later. And then next, like Yuki was mentioning before, quality assessment is really important. So we incorporated a quality assessment of the tissue. So as long as there's DAPI, Xenium will basically give you a cell output. But the trouble with, I keep saying the trouble with this, is that it's not necessarily in regions which are spatially preserved. So you would be analyzing things which aren't informative, whether it's crush or hemorrhage or tissue folding, etc. So given this, we also annotated, or pathologists annotated, regions which were spatially unpreserved. So you can see that in the blue polygon here. And then, like you would do on a pathology slide, we also annotated regions of excellent preservation, which is where we'll do our spatial analysis. And then we also incorporated a thresholding by transcripts, like you would normally do. So what we found was that I did transcript thresholding. It's like under 4, which you can see in the orange cells here, but it's not removing the regions where we know it's spatially unpreserved. And then the question is, well, why don't you just increase the transcript threshold? But then the trouble is you start losing your cells in the areas which we know are spatially preserved. So we found that we wanted to incorporate both of these two quality assessments. And then we basically have a nice single cell data set. So we did supervised and unsupervised using a new cell paper out, and then Azimuth as a reference data set to capture all of our cell lineages, and we can compare them on the H&E to check that they're what we think they are. And then for the final part of our analysis, our pipeline, we augmented our data set by incorporating features which we can only get from the H&E. So segmenting the bones, adding in the fat, adding in our megakaryocyte segmentation phenotypes, spatial preservation, and our fibrosis maps. So now we can see how all of the single cells fit into a larger context. And then this gives us way more than the 2 views of the bone marrow which started with, and we can start all on the same plane, and we can start unraveling how they all fit in together. So I'm just going to present a couple of preliminary slides of the questions that we're trying to think about. So first, even without the spatial context, we've got a nice single cell data set, where importantly with stromal cells as well. So we can see that the spanning the whole range of MPNs. So we can see that there already we can see a distinction between the ET and PV, which are more benign, clustering closer to normal. And then you've got your pre-PMF following into your myelofibrosis samples. And pre-PMF is a transitionary state. So we wondered if perhaps the dots there, which are overlapping with the MF, might have a correlation with fibrosis and stromal abundance. So we plotted this on the right here. We haven't found a nice correlation between the abundance, which suggests that it's not such a simple story, or we might need to go in a bit further on the hotspots, potentially. Next, one of the key things with MPNs is that they acquire their driving mutations really, really early in life, and then there's a mean latency between the disease progression. And one of the thoughts is that the bone marrow microenvironment might be important to producing the mutant cells. So given this, we wanted to use our bone segmentation and incorporating our fat to try and investigate the different bone marrow niches. So I just chose a very simple representation of the endosteal potential niche. So these are the blue cells around the bone. And I wanted to see if there was a change between our normal samples and our highest disease state, myelofibrosis. So at first glance, you can see there's a nice differential abundance in blast cells. So I've defined blast cells as cells which co-express CD34 and KIT. But then you can see that actually all of the cells are decreased. But then when I compare it to the log-fold change of the localization of all of the cells compared to just the blast cells, you can see that this is actually real, which is nice. And then in other parts of our analysis, we've been guided by what we can see. So we see these massive expansions of megakaryocytes in the erythroid cells in our myelofibrosis samples compared to our normal. So we wanted to try and characterize this or assess them quantitatively. So I've just defined clusters as any cells which are touching, and then I can create an artificial threshold and say, okay, these clusters above 200 or 20 are abnormal. And you can see there's a nice differential abundance between these. But the next thing to do will be to assess whether they're localized together because they come from a common progenitor, and that would be very interesting. And then another huge advantage of adding spatial transcriptomics with the accompanying H&E is we can now for the first time correlate gene expression to our megakaryocyte phenotypes and then move it back into the H&E tissue. So you can see here we're already capturing correlations with megakaryocyte phenotypes and gene expression. So GATA2 is a known marker, or overproliferation of GATA2 is a known marker of megakaryocyte expansion. So it'd be interesting to see where these cells are situated in the space now in a bigger cohort. So that's basically the idea, that we take information from our spatial transcriptomics and then we expand it back into our H&E routine diagnostic samples, which we have way more of, and then they'll start to feed each other. So thank you very much for listening. I just want to thank all of our collaborators, funders, highlight Roz, who was instrumental in creating all of this data set and driving hypothesis. And yeah, thank you very much for listening. And if you have any questions.
All right, questions for me?
Is there anything you guys do special for collection to preserve RNA and help with that? I mean, this is like the obvious one for bone marrow, right? 'Cause we can barely get like work on bone marrow alone.
Yeah,
Single molecule stuff.
Yeah, so I asked Roz before I went to this talk because I thought that was going to be a question. And she basically said, you need a good pathologist who can choose samples. She's a pathologist, so it might be worth talking to her and she can actually talk you through. She's very talented, choosing the best quality samples, basically, EDTA for decalcification.
So, essentially screening before.
Yeah, that's basically what she said, yeah. Sorry?
You don't know any assay to pre-screen your samples before running to?
No, not that I'm aware of, no.
All right, thank you, Emily.
Thank you.